Assalamu alaikum. Hello everybody. I hope you all people are fine and enjoying the conference. Once again, Sabarias here, MPhil scholar. I take this privilege to warmly all of you on the second day of third international conference on applied zoology 2020. Today, I will be the moderator of the session, which includes ecology, fisheries, wildlife, biodiversity, and nutrition. I will start this session with the talk of our first speaker, Dr. Syed Makhdoom Hussain from Government College, University, Faisalabad. He will talk about his research work, fish meal, alternative protein source in aquaculture feeds. Please, Dr. Syed Makhdoom Hussain.
Bismillah. I am Dr. Syed Hussain, Associate Professor at Department of Zoology, Government College, University of Hyderabad. The topic of my presentation is fish meal alternative protein sources and aquaculture feeds. First of all, as we know that the global population is estimated to be increased up to 9.7 billion in 2050, and about 50% more food will be required to meet the demand of this fastly growing population. As the human population is going to be increased continuously, so one of the most crucial challenges faced by all over the world is to find the means to feed the people. Almost more than 50% of the world population is suffering from the undernutrition or malnutrition. So the importance of the aquaculture can be observed in current scenario as it ensures a rapid and consistent supply of aquatic species for food to overcome on this drastic challenge. Aquaculture Aquaculture is basically the culturing of aquatic organisms including fish, mollusks, crustacean, and aquatic plants. Fish is basically acknowledged as a major nutrient-rich animal source feed. In this slide, we can see that Indian major carps are being cultured in Pakistan on large scale. In subcontinent, freshwater aquaculture is dominated by major carp species like catla, Labiohita, and Siranus mirigella. And in Pakistan, Indian major carps are being cultured in polyculture for maximum utilization of the feed sources in pond culture. Beside Indian major carps, there are also some carps being cultured like the Cyprinus carpio, which is also known as Gulfam. In Pakistan, it is basically an omnivorous fish and easily available throughout the Pakistan. It has high commercial value. Orychromis niloticus, also known as the Nile tilapia, freshwater habitat. It feeds on the plankton, especially the phytoplankton and benthic algae. It has fast growth rate, and Tinophragmodon idella, that is also known as a herbivorous. That is also a herbivorous fish. It is a cultivable species with other major carps like Catla, Labiohita, and Siranus mirigella. It is considered among the highly growing species in Pakistan. Hypothalamic smolitrix, that is commonly known as the silver carp. It is the fish of freshwater that basically feeds on the both phytoplankton and zooplankton. It is widely introduced in Pakistan for algal control and as a food source. Aquaculture success basically depends upon the economy. Basically, depend on the economy of the feed. Aquaculture is developing more efficiently than any other food production sector. However, economic factors such as the cost of the feed are limiting its development. As we know that feed primarily accounts for the 50 to 60% of the total cost in fish farming. So the main objective of the fish farmer is to produce high quality fish feed at low cost. In past or in previous days, the fish meal was being used on the large scale to prepare the fish feed. Fish meal basically is a grind or ground dried fish and fish waste used as a fertilizer and animal feed. It is being used as a major source of uh, feed for the aquaculture species like fishes. It is also serve as taste attractant for the herbivorous and the omnivorous fish species such as the Indian major carp and the Chinese carps. Fish meal was being used due to its number of benefits, like it has essential amino acid, it has a good profile of the fatty acid, it is highly palatable and contain high digestible nutrients like protein, fat, etc. Besides these benefits, this fish meal have some problems like its demand is continuously rising, whereas its supply is totally unpredictable. So making it more expensive. So to overcome these problems, we have to search for the alternative of this fish meal. And when we search for the alternatives of the fish meal, we find plant byproducts that are cost effective and environment friendly. Especially these are biodegradable, have potential source of the protein and energy. They are less in cost. They are helpful in developing environment friendly aquafeed. Here is some uh, examples of the plant byproducts that are being used commonly nowadays in Pakistan, like sunflower meal, cottonseed meal, canola meal, etc. First of all, we talk about the Moringa oleifera. As we know that the Moringa oleifera basically known as the miracle tree that have an impressive range of the medicinal uses. It is a highly digestible mm, uh, protein like 80 to 91%, whereas the undigestible protein ratio is 1 to 2% of the total. So protein contents is reported more higher than the soybean varieties. And when we talk about a moringa oleifera leaf meal, it is it have highly promising protein sources at low level. It is an important source of the vitamins and minerals in fish feed. It is also rich in carotenoids, ascorbic acid, and minerals, especially iron, that is very useful for fish health. It have high amount of the other minerals such as the calcium, copper, zinc, potassium, phosphorus, magnesium, and sodium, etc. And it have a crude protein value that is 25 to 32%. That is very suitable to formulate the fish feeds. And this next one is Moringa oleifera seed meal. In Moringa seed, we find about 34 to 38% protein, 8.57 percent crude fat, whereas a wide range of the essential amino acids, especially methionine, cysteine, and tryptophan, etc. It also has saturated and unsaturated fatty acids in good amount. And the next is canola meal, that is also a good protein source and because of a good amino acid profile. Is good source. It has good source of the selenium and phosphorus contents. Sunflower seed meal is also being used in Pakistan on large scale. It is a very commonly present all over the Pakistan. No doubt, when we use the plant byproducts, we have to face some problems or challenges that uh, presence of the anti-nutritional factors in plant byproducts such as the phytates, lectins, saponins, protease inhibitor, tannins, glucosinolates, oligosaccharides, etc. So we have to search to overcome these problems facing during the use of plant byproducts in fish feed. Various supplement may be added in the fish feed. That is the best solution to overcome such problems. And when we see about in the supplements, we have a large range of enzymes like phytase, protease, amylase, etc., and organic acid. Among organic acid, citric acid, very much famous, probiotics, prebiotics, nanoparticle like selenium and chromium, etc. First supplement in our list is phytase, that is basically known as the mannitol hexophosphate carbohydrolase. It has so many advantages when we use in fish feed. It improves nutrient digestibility and fish growth performance. It improves the minerals availability. It also increases the bioavailability of the phosphorus, and resultingly, it is very much helpful to develop cost-effective and environment-friendly fish feed. Here you can see a typical structure of the phytate or phytic acid, in which we can see that the yellow color indicating the phosphorus, that is very important mineral, but is chelated with other nutrients like carbon, hydrogen, and oxygen, etc. So there is a need of certain enzyme which can break down these chelated complexes easily to make these nutrients available to the fish. And the other supplement is organic acid like citric acid that modify the mineral transport mechanism by altering the pH of the fish stomach. It also decreases the pH of the fish feed. Citric acid has unique flavor and high buffering capacity, being used on large scale nowadays. When we in this diagram you can see that when we add citric acid in fish feed, it will lower down the pH of the fish feed, that resultingly will decrease the intestinal pH. When intestine pH will be decreased, then the efficiency of the phytase enzyme will be increased, and both phytase and citric acid will break down the chelated complexes of the nutrients. Resultingly, they will increase the bioavailability of the nutrients, and ultimately, we will have a good production of the fish. Supplement is the nanoparticle. As we know that the size of the minerals in nanoparticle is smaller than the 100 nanometers, so they may increase the bioavailability of the nutrients and minerals. In our lab, we have tested two major nanoparticle like selenium and chromium. Selenium is an essential trace element as it is involved in the enzymatic activity and prevention of oxidative damage to body tissue. The deficiency of the selenium causes great loss in livestock, and chromium is also involved in the carbohydrates, lipid, protein, nucleic acid metabolism. It is supplementation of the chromium nanoparticle have beneficial effects on the carcass characteristic, meat quality, and individual skeletal muscle weight. It also increases the tissue chromium concentration in selected muscles and organs. In this figure, you can see that when we added nanoparticles, especially at 2 milligram per kg, they improve the hematology of the fish by improving its quality. The probiotics are also being used. Probiotics are also microorganisms and substances that contribute to the intestinal microbial balance and microbial cell administration, which reaches the gastrointestinal tract and remain alive with the aim of improving health and growth. Nowadays, probiotics and prebiotics are being used on large scale in fish feed that improve the nutrient digestibility, and ultimately the overall production of the fish is being increased. And at the last, it seems that several authors working in the fish feed research have agreed on the inevitable requirement of using the plant byproducts based protein ingredient to replace fish meal in the diet for commercially cultivable aquatic animals. So, taking this board, we may hope that the fish meal will no longer be a part of the fish diets in future. Although there is a major challenge in the expansion of the plant ingredients, it is justified by many researchers that through proper dietary tactics, fish can be fed with excess plant protein without any negative performance. Use of plant byproducts may lead to a way for better coordination of fisheries with agriculture, and can achieve a goal of better acquisition in an eco-friendly, cost-effective, and more sustainable way. Thank you.
Thank you so much, Dr. Sayed Makhdoom, for such an informative talk. Now, moving towards our next presentation, I would like to call Dr. Sarish Periyal from University of Veterinary and Animal Sciences to discuss ND2 gene-based DNA barcode for genetic characterization of Pakistani pigeons. Please, Dr. Sarish Periyal.
Bismillahirrahmanirrahim. I'm Dr. Sarish Varal, working as Assistant Professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. Today I'm going to talk on one of the very important topic of the genetics, that is the use of the mitochondrial DNA for the genetic characterization of the avian species and particularly the Pakistani pigeons. And for this current research work, we have used we have choose very important mitochondrial genes, that is the ND2 gene. ND2 gene based DNA barcode for the genetic characterization of the Pakistani pigeon. First of all, the little bit brief introduction about the avian. Pakistan is actually blessed with a very diversified avian fauna. Among them, the pigeons are one of the most important usage bird. And if we talk about their identifications, so far phenotypic methods have been used, but there are some limitations. There are some pros and cons of the phenotypic phenotypic characterization and identifications of the bird. That's why we need some. There's a dire need of, and there's a need of the time for development of such barcodes, so that by using them we can characterize the birds in an authentic way. Now, this slide really shows a little bit introductions about the pigeon, what is actually the pigeon is, and this is the taxonomical information regarding the pigeons. Pigeons belongs to order Columbiform, and the family is Columbidae, and genus is Columba. And so far, there are 12 species in the uh, Pakistan that has been characterized phenotypically on the phenotypically by the birds breeders. And although by the fancier and the bird breeders, they have been there are some traditional and there are some classical approaches for their identification, like using their body weight, their shank length, their skin color, earlobe, the ear color, neck muffs, and the feather pigmentation. All these can be contributed to identifications for phenotypic identifications of the birds. But there are some problems that all these methods can be affected by some other factors as well. So all these methods are the crude one. That's why geneticians and the biotechnologists, they are in a search for the development and such authentic genetics. By using them, we can well identify the bird species. And among them, the molecular approaches, the rapidly amplified polymorphic DNA, restriction fragment and polymorphic DNA, microsatellites analysis like VNTRs and STRs, and mitochondrial DNA analysis, they are important one. And for this current research work, we selected the mitochondrial DNA. There are some characteristics of the avian mitochondria, that there are exactly 337 genes, and this is going to span over the 15 to 17 kb approximate genome length, and this is little bit different from the nuclear DNA. And as far as its inheritance pattern is concerned, it do not follow the classical Mendelian inheritance. The inheritance is exclusively from the maternal sides, and these genes are very important for the phylogenetic analysis as well, because there are certain regions in the mitochondrial genome which are highly conserved, and there are certain regions in the mitochondrial DNA, they are highly hypervariable. That's why it's one of the important, very useful milestones for the study of the phylogenetic studies. And there are, as it has been mentioned, that there are 37 genes, and among them ND2 mitochondrial gene is a very important, that is involved in the encoding of the enzyme NAD ubiquinone oxidase reductase chain 2. And this is a little bit conserved region as compared to the D loop, cytochrome B and CO1, and that's why this has been used for the identifications of the markers at a species and the subspecies and the breeds level. Phylogenetic analysis, for the phylogenetic analysis, again, this is a very important one. And Under these objectives, the study has been planned. The following coordinate objectives: the genetic characterization of Pakistani pigeons through mitochondrial antidote, their identification of their at their taxonomic level, and ultimately, finally, their phylogenetic analysis. And this is slide actually shows the methodology step by step by using that by following that we have conducted this research work. First of all, the birds have been selected on the basis of phenotypic characteristics that and then phlebotomy has been performed by the for the sake of the blood collection from the wing vein, and that is actually the brachial vein, and the DNA extraction has been done by using the organic because in the starting the blood sample was very low. And the primer has been designed for the amplification of the NG2 genes because mitochondrial full genome is available in the NCBI repository. And then PCR amplification was done, followed by the sequencing and bioinformatics analysis. This slide actually shows that how this is just we have to puncture the brachial vein, and then we can get the blood samples. And DNA extraction by using the, as already mentioned, by using the organic method, and DNA sequencing was also performed by using 3130 Excel genetic analyzers by ABM Prism. And following analysis has been done. Chromatogram have been studied through ChroMass. Homology analysis has been done by the NCPA BLAST. Multiple alignment of the sequences has been done by using the cluster W. And for the diversity analysis and the phylogenetic analysis, the PopGen and the MEGA5x2 was used respectively. And this actually shows the studies part. So far, we have studied the D loop, study from B and D2, but at the moment, the focus is on the NG2 genes. These are the different areas of the Pakistan from which we have done the extensive sampling for the sake of the different breeds of the pigeons. This is the Shirazi pigeons. You can see the characteristics on the basis of which they have been selected. The Lucky pigeons and the Little Rocks pigeons. And the sequence analysis of NG2 genes actually shows that there are some SNP has been identified that can be used as the DNA barcode markers at a different position. At the 2.218 position, there is a and a G patron chain, and there at the 817 position there is a G and a chain from the reference sequences. So this can be used as a molecular marker for identification. Same is at the Lucky and the Rock pigeons and the Shirazi and the Lucky pigeons. So these are actually the SNPs and the polymorphism which can be used for the development of the barcodes, and these barcodes can be can be used for the identification of their authentic identifications of the bird. So by using these marker, we can overcome the problem of the traditional approaches that can be employed for the identifications, and we can also develop the kit on the basis of these SNPs. And that can be very beneficial for the fancier and especially for the birds breeders. So thank you very much. This was for today's presentations.
Thank you so much, Dr. Sarish Priyal, for amazing talk. Now I would like to call Dr. Danish Riaz from University of Education to discuss the efficacy of probiotics on growth performance, nutrients, and mineral digestibility of Sir Highness Marigalla fingerlings, fed sunflower oil seeds byproducts, meal-based diet. Please, Dr. Danish Riaz.
Bismillahirrahmanirrahim. Third International Conference on Applied Zoology 2020. I'm Dr. Danish Riaz, Department of Zoology, University of Education, Faisalabad campus. The first presentations in international conference is efficacy of probiotics on growth, nutrients, and mineral digestibility of the Sir Highness Marigalla fingerling fed the sunflower oil seed byproduct based diet. Aquaculture is the fastest growing food producing industry in the world. The main objective of the global aquaculture is to maximize the efficiency of the food production system, development more efficiently than any other food production sector. However, the economic factors such as the cost of fish meal is limiting its effect of probiotics on the growth of nutrient digestibility, mineral digestibility of the Crinus megala fed the sunflower seed based diets. The probiotics added at the differentiated levels 0.5, 1, 1.5, 2, 2.5, and 3 gram per kg. The feces from each tank were collected twice a day, and the fingerling were fed at the 4% of live weight, and the measurement of the water quality parameter on daily basis. The fish meal is a healthier source of nutrients. There are following benefits. One is essential amino acids, fatty acids, and it's a highly palatable and contain the highly digestible nutrients. But there are following problems with the fish use of meal in aquaculture industry is a rising demand and unpredictable supply. It is a high cost and static level of production. These are the following problems in aquaculture industry. So we have to move towards an alternative source like the plant meals, and due to the digestibility problem, we have to move towards the probiotics. Probiotics are the micro organisms and the substances that contribute the microbial balance, and microbes are administered in a certain way which leads the gastrointestinal tract remain alive with aims improving of health and growth. And these are the growth performance of the Crinus megala on graded level used for the protection. Sunflower meal-based diets and the probiotics, which is commercially available in the form of protection, is used at the different graded levels 0.5, 1, and 1.5, 2, and 2.5, and 3 in various diets. We have noted that the optimum growth parameters are optimum at compared to the control, which is zero concentration of the probiotics. And it will also be noted that the growth parameters improve from the from 0 gram per kg to 2 gram per kg, and it will be decrease simultaneously and at the lowest at the 3 gram per kg. So the optimum level is 2 gram per kg for the in aspect of growth performance. These are the analysis composition of the apparent nutrients in the diet of Crinus megala, and the analysis composition of the feces of Crinus megala, and the nutrient digestibility of the Crinus megala fingerling. These are the apparent composition of the SFM-based diets and other test diets, and the optimum level is 2 gram per kg, as noted earlier. And this is analyzed composition of the in feces of Crinus megala fingerling for the SFM-based diets, control diets, and test diets supplemented with probiotics. And we have to note that improved at 2 gram per kg as compared to other graded levels. So the optimum level is the same in is 2 gram per kg as compared to the other graded levels. Conclusion. Results indicated that the maximum growth, nutrient digestibility, higher nutrients contents like protein, fats, cross energy, and absorption of the minerals of the Crinus megala fingerling were noted at 2 gram per kg level of probiotic supplemented SFM-based diet. It was concluded that the probiotic supplemented at 2 gram per kg level were helpful to develop a eco-friendly and cost-effective fish meal by using the oilseed-based diets. Thank you for your kind attention.
Thank you very much, Dr. Danish Riaz, for such an informative talk. Now, I'm very glad to call Sadia Tabassum from Government College, Unabad, to discuss the effects of Moringa oleifera leaf meal-based diet on the hematological indices in His Highness Marigalla fingerlings. Please, Sadia Tabassum.
My name is Sadia Tabassum from the Department of Zoology, Government College, University, Islamabad. My research title is Effects of Moringa oleifera leaf meal-based diet on the hematological indices in Spinous varicella fingerlings. Spinous varicella, commonly called mori, it is one of the Indian major calf cultivorous. It is a high consumer choice, widely cultivated under polyculture system. It has maximum utilization of food resources. Aquaculture it is the fastest growing food producing industry, developing more efficiently than any other food production sectors. Demand of quality protein, but have raised several issues that need to be addressed. One of these issues is development of fish feed from high quality, inexpensive sources, methods for making the fish free from anti-nutritional factors, cost of fish feed. These are economic factors that are limiting its development. Fish meal. It is a major source of protein and essential nutrients in fish feed. However, increasing demand, unstable supply, and high price of the fish meal with expansion of aquaculture made it necessary to search for alternative protein sources. Primarily accounts for 50 to 60% of the total cost in fish farming. The main objective for most fish farmers is to produce high quality fish meal at low cost. Increasing fish meal prices motivated the scientists to identify the cost effective alternatives of fish meal. Plant byproducts, which can be used as a replacement of fish meal, most promising sources of protein and energy for aqua feed. It is biodegradable, cost-effective, environment-friendly fish feeds. Moringa leaf meal, it is a promising protein source at low levels. It is important sources of vitamins and minerals, rich in essential amino acids, proteins, ascorbic acids, and minerals, especially iron, due to favorable amino acid profile and wide availability, MOLM to high amount of other minerals such as calcium, copper, zinc, potassium, phosphorus, magnesium, and sodium. The leaves contain full proteins 25% to 32%. It is a miracle free, has an impressive range of medicinal uses, highly digestible due to a high proportion of pepsin, soluble nitrogen 82 to 91%, and low proportion 2% of insoluble protein. Materials and methods, fish and experimental condition. Nice Marigella fingerlings were bought from the government fish seeds hatchery Kasaba. They were acclimatized to experimental conditions in laboratory for 2 weeks in V-shaped tanks. They were twice daily to appearance occasion on the vessel type used in the subsequent digestibility study. Aviation was provided round the clock to all the tanks through the Blu-ray system. Water quality variables such as water temperature, pH, dissolved oxygen were monitored. Processing of moringa leaf meal. Moringa leaf meal was obtained from the southern Punjab. They were soaked overnight in a tank to remove saponins and other water-soluble anti-nutritional factors. They were placed on a wide mesh to drain excess water and then spread on plastic sheets to dry under shade to avoid loss of vitamins through photodynamic damage or oxidation. They were threshold from stocks to reduce root fiber content in the meal, then grinded into a fine powder using a hammer mill and stored in plastic bags at room temperature. Feed ingredients and experimental conditions. Feed ingredients were analyzed for chemical composition. The reference diet was prepared to supply adequate levels of required nutrients for normal fish growth. Formic oxide was used as an enhancer. Formulation of pellets. The feed ingredients finally grinded to pass through 0.5 millimeter size. All ingredients mixed in a mixture for 5 minutes and fish oil gradually added there. 10 to 15% water added to prepare suitable dough. Processed by flooding machine to make feed pellets. This is a table which shows ingredient composition of test diets. Hematological study, firstly, anesthesia with low oil, aspiration of 6 grams per liter was given to Rosemary Gala for 5 minutes to collect blood samples from the caudal vein of fish. For analysis of hematological parameters, blood samples were taken to the molecular lab, Department of Biochemistry, University of Agriculture, Islamabad, Pakistan. The hematological parameters: red blood cells, hemoglobin, white blood cells, mean cell volume, mean cell hemoglobin, and mean cell hemoglobin consumed by using automated cell counter. Platelets were measured as described by Rahimi et al. The packed cell volume was measured after placing sealed microhemocrite tube in a centrifuge at 10,500 using a hematocrit feeder. MCHC, MCV, and MCH are found by drawing formula. This table shows hematological parameters. There are different levels of molecular-based test sites. The results of hematological parameters. The hematological indices of finger spread 10% amylin-based diet were found to be significantly different from the fish fat control diet. Red blood cells, white blood cells, hemoglobin, and mean corpuscular hemoglobin concentration of fish showed a significant inverse correlation with increase in amylin in diets, whereas platelets flowed by 2.47 in the fish fat on control diet, packed cell volume, mean corpuscular hemoglobin, and mean corpuscular volume in fish fat 10% amylin diets, which showed an increasing trend as amylin increased in the diet. Conclusion. It was concluded from the research work that MLM has a good perspective to use as FM regular diet up to 10% level without hematological disturbance, whereas conclusion of MLM above 20% in the diet showed the hematological disturbance. These are the following references which are used in my research work. Thank you for your attention.
Thank you very much, Sadiat Bassam, for interesting talk. Now I would like to call Zahidullah from Kohart University of Science and Technology to discuss the identification and characterization of Clostridium perfringens in the sheep and goats population of district Abbottabad. Please, Zahidullah.
Hello everyone, this is Zahidullah, a student of MPhil Zoology from Department of Zoology, University of Science and Technology, Cohort. The topic of my research is identification and characterization of Clostridium perfringens in the goats and sheep of district Abbottabad. Reported in USA, Iran, China, and Lahore in Pakistan. However, studies regarding Clostridium perfringens in the infected sheep and goats of Abbottabad, Khyber Pakhtunkhwa were not yet reported. For this, we designed a hypothesis, that is, Clostridium perfringens might be present and causing infection in goats and sheep of district Abbottabad. And our objectives from the study were to isolate Clostridium perfringens from the small ruminants, that is, goats and sheep of district Abbottabad, as well as to characterize the Clostridium perfringens isolated from the small ruminants of the study area. Material methods which we have used during these studies are ethical approval. Before starting our research work, we got ethical approval certificate from Ethical Committee of CUST. After getting ethical approval certificate from Ethical Committee of CUST, we started sample collections and collected a total of 256 samples from infected goat and sheep belonging to 8 different topographic areas of district Abbottabad. The samples were 2 gram fecals obtained through a sterile swab, which were transported to a laboratory in the Department of Animal Health, University of Agriculture, Peshawar. After processing the fecal samples, they were cultured in thiglycolate broth, and after culturing in thiglycolate broth, it were subjected to isolation on perforingens agar base. After incubating for 24 hours on perforingens agar base petri plates, a characteristic black color colonies were formed, which is the first identification mark of Clostridium perfringens. After bacterial isolation on agar, the inoculum was subjected to biochemical test, that is gram staining as well as gel liquefaction test. After confirmation through gram staining and gel liquefaction test, a stock was preserved and DNA was extracted. Once the DNA was extracted, it was confirmed by using species-specific gene, and the DNA was amplified through PCR, and the PCR product was confirmed on gel electrophoresis. These are the areas which I have collected the samples from infected goods and sheep of district Abbottabad, that is Kalandarabad, Sajikut, Havelia, Amirabad, Sherban, Sadikabad, Damtur, Kutiala. The red color graph show good sample, while the green one shows sheep samples. These are few of the pictures of agar plates. We can see the black color colony on perforingens agar base medium, gram staining, gel liquefaction test. This is the picture taken from agarose gel, and L represent the ladder, S represent the sample, N represent the negative control, while P represent the positive control. 204-7 base pair represent the amplified gene of alpha-toxin. Conclusion: Clostridium perfringens is highly prevalent among goat and sheep of district Abbottabad, found by this research work, which is indicated by the graph below. Total samples of sheep were 135, in which 41 were positive, while 94 were negative. There were 121 samples of goat, in which 28 were positive, while 93 were negative. Thank you all for your attention.
Thank you so much, Zahidullah, for your amazing talk. Now, I would like to call Noor Tariq from Government College University, Faisalabad, to discuss the effects of protease enzyme on growth and body composition of Sirhinus marigalla. Please, Noor Tariq.
Assalamualaikum, my name is Noor and the topic of my presentation is effects of protease supplementation on growth and body composition of Sirhinus marigalla. First of all, we'll discuss about the intro of our topic. Aquaculture. Aquaculture is one of the fastest growing food industry in the world. Asia is the major contributor. Future aspects of the agriculture can be based on the length of coastline and available land area in different countries. Pakistan ranked 34 number in the world for fish production. The next slide was also about the introduction of the topic. 7 warm water fishes and 2 cold water fishes are cultivated in the Pakistan. Fish feed, major source of protein. Expensive feed is one of the major problems in the aquaculture field. Alternative source of protein is the plant protein like soybean and sunflower meal. Soybean meal is potentially considered as the most important protein source as an alternative to fish meal. Soybean meal is characterized by the high protein content and a low fiber content. The next slide is about the why we choose Sir Highness Marigala. Sir Highness Marigala is the most important member of a carp family and ideal for polyculture. It tolerates the wide range of temperature, accepts the supplementary diet, delicious and very prestigious fish. Low cost of production, high market demand. These are the valid reasons why we choose Sir Highness Marigala. The next slide is related to the enzyme supplementation. What is enzymes? What is the basic means of enzymes? The basic purpose of enzyme is that they speed up the biological chemical reaction. They are also known as the catalysts. They convert the complex substances into the absorbable substances, reduce the anti-nutritional factors, increase the dietary energy, make the fish perform better. The enzyme which we are using in this experiment is the protease. Protease. The main function of the protease is that it increases the absorption and digestibility of the nutrients like fats and proteins. Animals start taking more food due to which weight increases. It also enhances the metabolism. Ammonia production in the water reduces due to which the water clarity level increases. Know the materials and method. In materials and method, first of all, we will discuss about what is our experimental fish. The experimental fish is the Serhanis marigala. The single use of the Serhanis marigala was purchased from the fish seed hatchery Satyana Road, Islamabad. The duration of the experiment is 19 days. Before the start of the experiment, fish were acclimatized for the 2 weeks, feeding on a controlled diet. 4% live body weight in a glass of feeding. The next is the feeding protocol. After acclimatization of 2 weeks, fingerlings were transferred into the glass aquarium for each treatment. 2 replicas were used, and in each replica, 20 fingerlings were stocked. Water quality parameters like alkalinity, pH, and the water temperature were monitored. Dissolved oxygen, which is 5 to 7 ppm, were maintained using air pump propelling system. The next one is the feed preparation method. The feeds were prepared by the linear formulation method. Feeds were prepared by mixing the appropriate amount of all the ingredient and enzymes. Fish oil was added while mixing was continued for further 5 minutes. 10 to 50% water was added to prepare a rough dough and then dried and stored at minus 4 degrees until use. The next table is the formulation of the feed table. Next, we will discuss about the experimental trial with growth parameter. During the trial, fingerlings were taken from each replicate on the fortnight basis to determine the growth performance. The main formula which is used for this specific growth is the final net body weight minus initial net body weight multiplied by 100 over total time duration. The next parameter is the analytical analysis. Analytical analysis including the ash analysis which were done by the electric furnace, dry matter which is done by the oven, and the crude protein which is done by the microjet apparatus. Crude protein is done by the chloroform methanol extraction method. The next we will discuss about the results. In the results, the first parameter is the growth parameter. As you see, this is the graphical representation of increasing rate of Serhanis marigala based on the control and experimental diets. The red line shows the maximum increase in the body weight. The red line indicates the treatment PT1. PT1 is the treatment that has the protease 0.1 mg/gram. The next is the body composition. This is the graphical representation of the dry matter and crude protein of Serhanis marigala rare on the control and experimental diet. Dry matter shows the maximum value in the T2 treatment, whereas the crude protein also shows the maximum value in the T2 treatment, which is the protease 0.2 mg/gram. The next one is the graphical representation of crude fat of Serhanis marigala rare on the control and experimental diet. The maximum value of the crude fat was shown in the PT1 treatment, which is 0.1 mg/gram of the protease. We concluded from this experiment that the protease supplementation in the fish feed improves the growth and the body composition of the Serhanis marigala. Thank you very much. stop sharing में हो गया अभी.
Thanks a lot, Mariam Tariq, for your amazing talk. Now, moving towards our next presentation, I would like to call Mariam Yasin from Agriculture University, Faisalabad, to discuss the zoological gardens, a research gate for the conservation of wildlife. Please, Mariam Yasin.
Bismillahirrahmanirrahim. I am Mariam Yasin and I have done my master's degree from University of Agriculture, Faisalabad, Pakistan. And today I'm going to discuss about the zoological gardens as a research gateway for the conservation of wildlife. So first of all, we have to take a look about the idea, what are zoological gardens? Zoological gardens are those places that have collection of animals that are confined and are given proper habitat and food and are proper area, and they are protected under so basically, we can say that zoological gardens are the places that are meant to conserve the animals that tend to extinct drastically. So it is as it is the century of extinction, and the climate conditions and the other things are very drastic for the animals and all the living beings. So it is need of the hour to have such places that can provide animals with the proper conditions and other things which may contribute for the betterment of biodiversity. And the extinction rate is really drastic in this decade and in the recent years. So here are some extinct species in recent decade that are Western black rhino that has gone extinct in the recent past in the year of 2011, and it's been extinct due to the poaching and many other factors that contributed as like the habitat loss and human involvement are the basic reason. And the second animal is Spix macaw. It's been also just got extinct from the wild. And the third one is splendid poison frog, and it's also been declared as extinct in the year of 2020. And the reason for its extinction was chytis fungus outbreak. And the fourth one is winter giant tortoise, and it's been um, extinct from the environment. And the last one is Beiji, and it's a fish from China, and it has been declared very critical in the year of 2002. And then after that, it was not been seen in the wild for a long time, and it was just thought to be extinct from wild. But in 2016, some scientists just made a statement that they have seen this fish in the wild, and it was though it was not captured on cameras, but the scientists are saying that it is present, and it's under observation that either it is present or not. And the second thing is the efforts that are taken by the government and NGOs to make the extinction rate less. So the efforts of government and non-government agencies are really appreciable to just contribute in the period where things are going in a drastic manner. And here is some example where the Pakistani government has taken some steps for the conservation of wildlife, and it's about some national parks of Pakistan where different kind of animals and their species are held and captive in a proper condition, and they are just um, are allowed to grow in number. Like in the Osai National Park, brown bear has been captive and it's been breeding there for its increase in number. And the second major aspect of zoological gardens is ex situ breeding methods, ex situ and research perspective that has been taking place in zoological gardens. As we know that ex situ breeding methods are really very helpful for those animals that are going to be extinct or are on a verge of extinction, as it contribute to increase their genetic diversity and just assist the breeding of animals in their captive breeding. And Exito methods are really a hope to flourish again, motto, as they help the animals to just increase their numbers, even if their conditions are not proper in the wild. So, and man is making some effort to help those animals that are struggling for their survival in the wild. By just making some exito breeding method techniques. And secondly, ex situ methods are very helpful in the research perspective, either the genetic material that man or are more favorable for the survival of animals, and the genetic breedings can also be included in ex situ breeding methods. And here is the second thing, that is the contribution of zoos and aquariums in the reintroduction of animals into their natural habitat. That is the efforts taken by different levels of mankind and the animal as well. So it's been a percentage of zoos and aquariums where the animals are being reintroduced into their natural habitats. And there are many success stories regarding the reintroduction of animals in their natural habitats. For example, the number of snow leopard that had been drastically decreased, but now it's been more in number because of the efforts taken by the government and the non-government organizations. And so the number of snow leopard has been recovered from critically endangered to endangered, and we hope so that it will make a proper number, that we won't make any effort to just to make any kind of alarming calls for people. So these are some success stories that we can relate with the reintroduction and rehabilitation of animals in their natural ecosystems. So we are concluding that if we are going to enjoy the thing as ourselves, then why we won't make that for our next future generations? So it's our duty to make some efforts to make the things as possible and great for our future generations. So we have to make some extra effort. These places there where animals are kept and are reintroduced into their natural environments, so that our future generations can see what we have seen in our lives, and they can also enjoy what we have enjoyed in our lives. Thank you.
Thank you very much, Mariam Yasin, for your nice presentation. Now, I would like to call Muhammad Zubair Al Hassan Arslan from Government College University, Faisalabad, to discuss the effects of phytase and citric acid supplementation on growth performance and nutrient digestibility of cyprinus carpio fingerlings fed on canola meal based diet. Please, Muhammad Zubairul Hassan Arslan.
Bismillah Rahman Rahim. My name is Muhammad Zubair Hassan Arslan. I am PhD scholar. My topic of research work is effect of phytase and citric acid supplementation on growth performance and nutrient digestibility of Saprinus carpio fingerling fed on canola meal based diet. Aquaculture is developing more efficiently than any other food sector. However, economic factors such as cost of fish feed are limiting its development. Aquaculture feed industry mainly depends on the use of fish meal due to its high nutritional and palatability value. Fish meal is a essential amino acid and fatty acid required of the optimum fish growth. Plant byproducts are the promising source of protein and energy and may be used for formulation of cost-effective and environment-friendly aquafeed. Canola meal is a plant byproduct used in my research work. Canola meal is a valuable protein source for use in animal feed and it's contain 36 to 39% protein. Its amino acid profile is similar to that of herring meal protein and superior to soybean meal. Canola meal is being used up to 20% inclusion level of fish diet, but it is desirable to further replace fish meal in the diet due to the limiting world supply and growth demand for these highly valued species. This is a view of canola meal. Canola meal, the evaluation of the feed ingredient is the crucial to nutrition research and feed development for aquaculture species. Canola meal is a fish diet. Canola meal is a well established as a feed ingredient in salmon, trout feed, fish. Canola meal is used at up to 20% inclusion level in the salmon diet. This is the first supplement which is used in my research work, phytase supplementation, and the second one supplement of use in my research work is citric acid. Citric acid decrease the pH of digestive system, stimulate feed consumption that will result to better absorption nutrients. Supplementation of the short chain organic acid, citric acid, unique flavor and high buffer capacity. This is the Saprinus carpio fish, which is used in my research work. Saprinus carpio is commonly known as the gulfarm, bottom feeder, high consumer choice, widely cultured under the polyculture system. Material and methods. Fish and experimental occasion. First, the fish were purchased. Fingerlings were purchased from fish feed hatchery and treated with NaCl solution, acclimatized for 2 weeks, continuous aeration, water temperature, and pH measures. This is the composition of ingredient composition, feed formulation. Firstly, the ingredients purchase, analyze, grinding, and electric mixture and mixing. This is the inner view of fish tank. This is the bottom view of fish tank. Calculation on nutrient digestibility on this formulae. Chemical analysis of feed and feces. Firstly, the feed and feces sample homogenized with the help of pistol and mortar. Crude protein directed by geldol. Crude fat directed by electric furnace. Crude fat detected by petroleum ether, ash detected by electric furnace, gross energy is detected by bomb calorimeter. This is a three-way ANOVA used in my research work. Result of the present work conducting the maximum digestibility on the T12 diet, which have 2.5% citric acid and 750 FTU of phytase enzyme. And the second highest Second best result were obtained by using the test diet T11, which 0% citric acid and 750 F2U of phytase enzyme. The high percentage of digestibility value of crude protein 77.38, crude fat 85%, and gross energy 70.47. T12 diet having 5% citric acid and 750 FTU of phytase. This is the growth table. This is the nutrient digestibility table. Crude fat, crude protein, and gross energy. This is the apparent crude protein and feces, crude fat, and gross energy. Conclusion. In conclusion, that the recent studies provides evidence to the citric acid and phytase supplementation chloramine based diet fed the Serpinus carpi fingerlin increase the nutrient digestibility and without any negative effect of the carcass composition and nutrient digestibility composition. Thank you. छोटे यार लोग
Thank you very much, Muhammad Zubairul Hassan Arslan, for your informative talk. Now, I welcome Ms. Banur from Government College University, Faisalabad, to discuss the study on growth performance, carcass composition, and nutrient digestibility of C. carpiojuveniles fed on Chenopodium quinaceid meal-based diet. Please, Ms. Banur.
Bismillahirrahmanirrahim. Assalamu alaikum. My name is Miss Banur. I have recently completed my MPhil Zoology degree from GC University, Faisalabad. I am presenting the topic Study on Growth Performance, Carcass Composition, and Nutrient Digestibility of Oreochromis niloticus juveniles fed on Chenopodium quinoa seed meal based diet. Aquaculture is an important part of our food production industry. The world population is increasing so fast as compared to the food sources, due to which there are the needs to develop the strategies for fulfilling the demands of food. The cost of fish meal and some other factors affecting the development of aquaculture industry, which limits its profitability. In fish farming, feed accounts for 50 to 60% of total fish farming cost. The primary goal for most of the fishermen is to produce high quality feed at lowest prices. Plant-based protein sources of feed are suitable and profitable for aquaculture. Increasing prices of fish meal motivate researchers to find cost-effective fish meal alternatives. Chenopodium quinoa is a member of the group of pseudocereals due to its good amino acid profile and unsaturated fatty acids, vitamins, dietary fibers, and minerals. Quinoa has excellent nutritional values. It was described as the most nutritious grain in the world by Smithsonian Institute, Washington. Total protein content of quinoa seed is 16.3% dry basis, greater than that of the barley 11% dry basis, rice 7.5% dry basis, wheat 15.4% dry basis. Quinoa becomes a high quality feedstuff due to its relatively high protein, fat, and starch contents. Methionine and lysine content in quinoa is 40 to 100 and 510 to 640 milligram per 100 gram respectively, due to which it is considered as complete protein. It is reported that lysine content of quinoa is almost double that of wheat and corn and 25% higher than rice. The fish species I was chosen is the Nile tilapia, which is a sea-chilled fish native to Africa, but today its major aquaculture industries are in Asia. Supply of freshwater fish species, especially common carp, is well dominated in the aquaculture industries. Tilapia are the third most farmed fish in the world after the carps or salmonids. The current research work was carried out to search out the best and lowest cost protein sources such as quinoa seed meal for commercially important species that is Onoloticus to improve its production and to overcome the difficulties of expensive fish meal. Onoloticus juveniles purchased from Government Fish Seed Hatchery Faisalabad, acclimatized to experimental conditions in fish nutrition laboratory for 2 weeks in V-shaped tanks. Aeration round the clock is provided to all the tanks through capillary system. Water quality parameters such as water temperature, pH, dissolved oxygen were maintained. Seeds were dehulled and then grind to a fine powder and mixed with fish oil. Then 10 to 15% distilled water is added to make a suitable dough. After that, proceed by pelleting machine to make feed pellets. Weight gain percentage is measured by the following formula, and FCR is measured by this formula. Calculation of digestibility is measured by this formula. The fish is fed at 5% live wet weight. 15 juveniles were kept in each tank and triplicate tanks were used. Tanks were washed after the 2 hours feeding trial to remove all the uneaten particles of diet. Feces collected from the fecal collecting tube by opening valve 1 and valve 2. The feces are oven dried and analyzed by standard methods. Chemical analysis moisture determined by oven drying at 105 degrees centigrade for 12 hours. Crude protein by microgeldal apparatus. Crude fat by petroleum ether extraction. Ash by ignition at 650 degrees centigrade for 12 hours in electric furnace. Gross energy by oxygen bomb calorimeter. The results obtained in my research work shows that quinoa seed meal based diet enhanced the feed quality and increased the weight gain of fish. In Onoloticus juveniles, the maximum weight gain 13.65 gram was observed at 20% level of fish meal replacement with quinoa seed meal, and the value was significantly different from weight gain 11.95 on 0% level. At 50% replacement level, weight gain was 12.27 was observed. The value of FCR was 0.98 of Niloticus on test diet with 20% level. The graph is showing the growth performance, weight gain in grams, nutrient digestibility. The result of current research indicates that at 20% level, the rate of digestibility of protein was highest and different from all other test diets. It was also observed that 20% replacement level of quinoa seed meal, the crude fat and gross energy showed highest values which were significantly different from the other test diets. Carcass composition at 20% level, maximum values of crude protein, fat while the minimum ash content, moisture, crude fiber and carbohydrates were observed as compared to the other levels. The conclusions are all the results showed the replacement of fish meal with quinoa seed meal not more than 20% is the best level for maximum fish growth. Thank you.
Thank you, Ms. Banur, for superb talk. Now, I would like to call the second last speaker of the session, Hidayatullah Khan from Kahar University of Science and Technology, to discuss the genetic variations among different subspecies of Lodakkia pakistanica from different localities of Pakistan. Please, Hidayatullah Khan.
Bismillah ar-Rahman ar-Rahim. First of all, I would like to thank you, Organizing Committee ICAS 2020, for providing such a great opportunity to share my research work with scientific community. My name is Hidayatullah Khan, Department of Zoology, Kohat University of Science and Technology, Kohat. Introduction. A gamete is morphologically and ecologically a diverse sauroid family consisting of more than 330 contemporary species in 54 genera. This is the fourth largest lizard family and is widely distributed throughout various regions. A gametes occur in a wide range of different habitats including arid, semi-arid, tropical and subtropical regions. The genus Lodakkia comprises about 20 species mainly occurring in highland and mountainous regions of the Central and Southern Asia. This species inhabit whole surfaces and rocky structure near water streams and around human habitation and has been observed to hibernate in these surfaces during winter. In the western Himalaya, it can be frequently spotted basking on the rocks and wooden logs and on the thatches of houses and shops near human habitation. Lodakkia pakistanica have been observed active during warmer times of the day and are not sighted on days with low temperature and rains. It is an omnivore and its food mainly comprise of insects like ants, small arthropods, and lepidopterans. Lodakkia pakistanica, which has been described from Pakistan as Agama pakistanica, is widely distributed along the river Indus from Hunza, Gilgit, Chilas, Jaun to district Mansehra, Pakistan. The study records show four distinct populations, two of which represent new intraspecific taxa, whereas the fourth exhibit intermediate characteristics of two forms identified. The three subspecies of Lodakkia pakistanica are Lodakkia pakistanica khanai, Lodakkia pakistanica afanbargi, and Lodakkia pakistanica pakistanica. Objectives. Identification of Lodakkia pakistanica from northern areas of Pakistan. To find out genetic variation among the subspecies of Lodakkia pakistanica from northern Pakistan by using mtDNA gene ND4. Material and methods. Ethical approach. The study protocol was approved from the Research Ethical Committee of Kohat University of Science and Technology, Kohat. Sampling and study area. Approximately 97 specimens of Lodakkia pakistanica Lodakkia pakistanica was collected across its range in Pakistan, including parts of Mansehra, Kohistan, Chitral, Gilgit, Hunza, Skardu, Chilas, and district Swat. Sampling procedure. Live specimens were collected through bare hand using safety gloves. The specimens were euthanized by injecting absolute ethanol in the eye. Blood tissue in minute quantity were taken from each specimen in absolute ethanol and was stored at -20 degrees centigrade in a molecular parasitology and virology laboratory department of zoology cast cohort for further processes. Specimens were permanently preserved in 96% ethanol cataloged and stored at taxonomy laboratory department of zoology cast cohort. DNA extraction. DNA were extracted from the blood sample using the Coagene DNA extraction kit with provided protocol. PCR amplification and sequencing. Extracted DNA was subjected to PCR for amplification of mtDNA gene encoding NADH dehydrogenase subunit for NADH NAD4. An amplified product will be sequenced, and sequenced alignment was analyzed through bioedit, and MAGOX will be used for the phylogeny interpretation, species identification. About 20 samples were collected from Mansehra, 12 from Kohistan, 17 from Gilgit, 11 from Chitral, 12 from Hunza, 10 from Chilas, and 15 from Swat. And the total number of samples is 97. Prevalence of Ladakia pakistanica subspecies in different area of Pakistan. Ladakia pakistanica at Mansehra 0, Kohistan 0, Gilgit 13, Chitral 11, Hunza 8, Chilas 10, Swat 0, and the total number is 42. Ladakia pakistanica Khanai at Mansehra 18, Kohistan 12, Gilgit 0, Chilas 0, Hunza 4, Chilas 0, and Swat 7, and the total number is 41. Ladakia pakistanica Offenburgi at Mansehra 2, Kohistan 0, Gilgit 4, Chitral 0, Hunza 0, Chilas 0, Swat 8, and the total number is 14. This one is Ladakia pakistanica. This is Ladakia pakistanica Khanai, and that one is Ladakia pakistanica Offenburgi. At the end of this talk, I would like to thank you all for your time and for your attention. Thank you.
Thank you very much, Hidayatullah Khan, for such an amazing talk. Now, I would like to welcome our last speaker of the session, Syed Mohsin Shah from Kahar University of Science and Technology, to discuss the phylogeny of Ladakia tuberculata reptilia agmenta from northern Pakistan. Please, Syed Mohsin Shah.
Bismillah ar-Rahman ar-Rahim. First of all, I would like to thank the organizing committee, ICAST 2020, for providing me such a great opportunity to show my research work with the scientific community. My name is Syed Mohsin Shah, and I am MPhil Scholar of Department of Zoology, Kohat University of Science and Technology. And the topic which I'm going to present in this to the scientific community is the phylogeny of Ladakia tuberculata, that is the reptile Agamidae, and it is belong from Agamidae family, and the research area of my topic is northern Pakistan. So actually the topic theme of my presentation is phylogeny of Ladakia tuberculata from northern Pakistan. So here is the introduction that the genus Ladakia comprise about 20 species, mainly occurring in highland and mountainous regions of the Central and Southern Asia. And Agamidae lizard Ladakia tuberculata is commonly known as Kashmiri rock agama, and it is mainly distributed in northern Pakistan areas, Nepal, India, and it is the species is known as for its beautiful coloration, adaptability, and instincts with its foraging activity. This species inhabits holes, surveys, and rocky structures near water, stream, and around human habitation, and has been observed to hibernate the surveys during the winter season. The object of my research topic is to the identification of Ladakia tuberculata from northern areas of Pakistan and to elucidate the phylogenetic position of Ladakia tuberculata. So this is the overall overview of my materials and method. So first of all, in our research, we did the ethical approval pro forma, and then the sampling and study area. So sampling by we collect these different blood samples of Ladakia tuberculata from northern Pakistan with bare hands and safety gloves. So we collect a small amount of blood from their mouth in the blood tubes. And then for further process, we brought them into the laboratory of Cohort University of Science and Technology, and the laboratory name is Molecular Parasitology and Virology, a laboratory of Department of Zoology for further molecular work. After then, we extracted DNA from the bladder by using a Chiagen DNA extraction kit, and then further we amplify them for we send them into for PCR machine for amplification of PCR, and for sequencing, and for after that further result will be proceed, and the species identification well done were done. And this is the overall demography of my research work. We have collected almost 115 blood samples from different area of Pakistan, including a different area of northern Pakistan actually. And we collected 45 samples from Azad Kashmir, 37 from Kamrat area, and 33 from Mansehra. So this is the overall demography of our research work. These are some pictures that we collected from different areas of Pakistan. So the first picture is from AJK, Azad Jammu and Kashmir. The second picture is from the Kamrat Valley of KPK. And the third picture is from the Mansehra area. So this is the overall phylogenetic tree for this species of Lodacea tuberculata, and the evolutionary history was inferred using neighbor joining method. The optimal tree with the sum of branch length 12.9995978 that is shown into the picture as well, and the percentage of replicatory in which the associated taxa corrected together in the bootstrap test. 1500 replicates are shown next to the branches. So the tree is drawn to scale with branch length in the same unit as those of the evolutionary distance used to interpret the phylogenetic tree. So actually I did a work of phylogeny, and it is a type of work like molecular taxonomy of this species. So I did actually the molecular taxonomy of this species because the molecular work is all together lacking from Pakistan. Very less people use this type of work, and I did it from. So thank you so much for your attention and time, and once again thank you so much of ICAST 2020 for providing me a great opportunity to represent my research work with them. And do he, sir?
Thank you very much, Sayed Mohsin Shah, for a wonderful talk. And here are the talks and the presentations of Third International Conference on Applied Zoology 2020 has ended. I would like to say thank you to all the respected teachers, researchers, and dear fellows on the behalf of Applied Zoological Society of Pakistan. I hope you all people had a good time with us, enjoyed all the sessions, and learned a lot from informative talks. Once again, I would like to say thank you to all the speakers for delivering such an informative talks to our audience. And here will be our closing ceremony. Gold medals and awards should be announced. Stay tuned with us. Thank you very much.